So we are already recording the webinar and it's open.
Yes, I have just started the session, and now we have the participants joining us.
That's perfect.
Hi, can you hear me again? Super.
Yes, we can hear you, Bert.
Okay, perfect.
Hello to the people that is already connected. You can choose the interpretation language of your choice by clicking in the bottom part and select English, French, and Spanish. Bueno, buenos días a todos. Esta sesión de webinar tiene interpretación al inglés, a francés y español. Y ustedes pueden seleccionarla en la parte inferior de la pantalla. Good morning, good afternoon, and good evening. This workshop series, this session counts with the simultaneous interpretation into English, French, and Spanish. Please select the language of your choice in the bottom part of your screen. Buenos días, buenas tardes, buenas noches a todos. Este webinar cuenta con interpretación simultánea. Por favor, seleccionen su idioma en la parte inferior de la pantalla. We will start in a couple of minutes. Thank you for being on time. Before the start, please note that you can provide questions and answers through the Q&A channel, also in the bottom part of your screen. And if you have any difficulties, please write a message through the chat box. Okay, if everybody agree, we can start this webinar session. I would like to confirm if Mr. Osama Lisi is available to start.
Well, thank you so much, and good day, everyone. I want to thank everyone for taking the time to participate in today's session. So welcome, everyone, to the first session of this series of workshops on Fusarium TR4 diagnostics, surveillance, inspection, and simulation exercises. My name is Osama Elisi. I am the new secretary of the International Plant Protection Convention. And again, I want to thank you for joining us to this very important virtual workshop. I hope everyone is staying safe and healthy. And as you may know, the IPPC's mission is to working with everyone, all contracting parties across the world. We work to prevent the spread of plant pests and diseases globally through safer international trade. So the international standards for phytosanitary measures, or ISPMs, provide a set of global standards and common understanding towards achieving the purpose of safe trade. However, we know that the risk can never be reduced to zero. So it is incumbent upon all of us, all countries, to prepare and be able to respond effectively to pest outbreaks, as well as to the introduction and spread of emerging pests. This is why it is important, that was important enough that the IPPC strategic framework that was put together, adopted by the CPM from 2020 to 2030, the development agenda of the strategic framework incorporates specific item on strengthening pest outbreak alert and response systems. As many of you know, recently, two pests of primary concern to the IPPC community at large around the world, and have enticed global interest, effort, and activities to help minimize their impact. One of these two pests happened to be the fall armyworm, which is a transboundary pest. And the second one is the subject of today's session, is Fusarium banana wilt, also known as TR4. Last year, the IPPC Secretariat delivered a webinar series and corresponding guidelines on fall armyworms prevention, preparedness, response, recovery, and following the same line, the IPPC Secretariat is coordinating activities on Fusarium TR4, which is currently the most severe threat to banana production. Over the past 10 years, TR4 has made an intercontinental jump from Southeast Asia to Africa, 2013, and South America in 2019, basically representing some of the largest banana production areas in the world. This makes it even more significant that we prevent further introduction and spread of Fusarium TR4, but also once it's introduced, it is really important for us to minimize the impact by containing, suppressing, and managing the infection. So this workshop series was initially conceived under the framework of the project FAO Support to COMESA Trade Facilitation Program for COMESA countries. However, considering the threat that Fusarium TR4 represents, it was extended to 2 countries in Asia under the IPPC China project, and globally by including all interested regions and countries where Fusarium TR4 is or may be introduced as a quarantine pest. I take this opportunity to thank the COMESA Secretariat for their support and very positive role in executing the COMESA project and all activities that IPPC Secretariat is leading. This includes developing contingency plan guide and e-learning courses in export certification, surveillance, and PRA. I would also like to thank the COMESA Secretariat in advance for the support they will provide to conduct simulation exercises in 2 COMESA countries. The IPPC has developed several resources and materials where countries may find guidance to prepare and respond to pest outbreak, such as the surveillance guide, the pest risk communication guide, the pest status guide, among other resources currently available on the International Phytosanitary Portal. This workshop series also counts on the support of the Implementation and Capacity Development Committee, or the IC team on Fusarium TR4, which was created to help addressing the global issue of Fusarium TR4, and is composed of remarkable worldwide experts. I want to take this opportunity and thank each and every one of you for volunteering your time and contributing with your expertise, helping the whole world dealing with this very invasive pest. So I want to thank you so much. Today's session includes simultaneous French interpretation provided by Collectif, Comité de Liaison Europe, Afrique, Carib, Pacific, if I'm saying this correctly. So the Collectif, I wanted to thank you so much for your support. Also, we have simultaneous Spanish interpretation provided by SELA. And so I'm glad to acknowledge both institutions for their kind contribution and support. So in summary, I hope today's session provides you with a clear understanding of the relevant information on Fusarium TR4 diagnostics. And I wish you all very fruitful workshop. And again, IPPC stands ready to support you in any way we can, so we can really come up with a plan to addressing this issue in the most meaningful way. Thank you so much. Appreciate it.
Thank you very much, Mr. Elisi. This is an excellent introduction to our work and what we'll discuss within this webinar. I think Mr. Elisi will leave the workshop because he has another conflict meeting. Welcome again to the IPPC workshop series on Fusarium TR4 surveillance, diagnostic, inspection, and simulation exercises. My name is Camilo Beltran Montoya, International Phytosanitary Specialist within the Implementation and Facilitation Unit at the IPPC Secretariat. I will be the moderator of this first session of 3, and I take the pleasure to note that we have around 660 people registered from 116 countries. We count on the attendance of the National Plant Protection Organization's representatives, researchers, academics, and international organizations. So please introduce yourself in the chat, indicating from where you are following this webinar session. Thank you for the people who already did it. Now, I will take the floor to provide you with an insight on what is being done within the IPPC Secretariat and the content of the webinar workshop, and I will provide a brief reference to the COMESA project. So let's start with this webinar session, and may you please share the screen, Eva, with the presentation number 1. Okay, let's start. I will take you very quickly through the context of the emerging pests under the IPPC framework. Next slide, please. And the issue related with the emerging pests were merged with the activities to address the 2020 and 2030 IPPC strategic framework development agenda item on strengthening pest outbreak and alert response system. The Commission on Phytosanitary Measures created a focus group on pest outbreak alert and response systems that have made several recommendations that may impact how the IPP community deals with emerging pests. So far, 2 pests of primary concern for the IPPC community have been the subject of the global efforts and activities. As noted by our secretary, these two pests have been incorporated into the IPPC Secretariat work plan. These pests are fall armyworm, Spodoptera frugiperda, and Fusarium R4. Regarding the fall armyworm, the Technical Working Group on Quarantine and Phytosanitary Measures for Global Action on Fall Armyworm Control produced the IPPC Prevention, Preparedness, and Response Guidelines for Spodoptera frugiperda, which were published in Arabic, English, French, and Russian. In addition, the IPPC Secretariat, with the solid support from the Technical Working Group on Fall Armyworm, delivered in 2021 the webinar series, The Fall Armyworm, a global threat to prevent. And if this information, if this of your interest, you may visit the IPPC website and consult the guidelines and access to the recordings of the preparedness, prevention, and response guidelines for Spodoptera frugiperda. Next slide, please. So far, the IC team of Fusarium TR4 has completed a review of contributed resources that are posted on the IPPC webpage, is developing prevention, preparedness, and response guidelines for Fusarium TR4. And the IC team of Fusarium TR4 provided full support to organize this workshop series. Another relevant point to note is that a questionnaire to assess the country's capacities to respond to an outbreak of Fusarium TR4 will be launched very soon. Next slide, please. As noted by Mr. Osama during his welcoming remarks, this workshop series was initially planned for the COMESA project. And also, I would like to note that all the available guides and training materials are available in the IPPC webpage. To consult them, you can visit the IPPC webpage. This page contains many resources, including brochures, fact sheets, guides, and advocacy and training materials and studies. To consult them, you can go to the IPPC website, click on the tab Resources, and then select the training material box of your interest. Next slide, please. So, considering that the Fusarium TR4 is a global concern, this workshop series was extended globally, and because it's a concern of global interest. I'm going to provide you some insights of the COMESA project. COMESA is the common market of Eastern and Southern Africa, and the project aims to strengthen the capacities of the COMESA countries. The IPPC Secretariat supports this project by overseeing the creation of a regional networking platform for sharing information on risks to plant health arising from pests and disease, and the establishment of an early warning and emergency response system to facilitate collective actions in mitigating priority risks. Next slide, please. The IPPC and the COMESA Secretariat have been working to achieve the expected outputs of the project, which will include e-learning courses on pest risk analysis, export certification, surveillance, and inspection. Also includes guides on contingency planning and the conduction of simulation exercises for 2 selected countries in the COMESA region. And subject to the funds availability, is planned to strengthen the diagnosis capacities in the COMESA countries. Next slide, please. The content of this workshop series on Fusarium TR4 consists of 3 sessions to cover diagnostics, surveillance, and early warning, and inspection and simulation exercises. All of you are invited to register for each session. Next slide, please. Now, the objectives of the workshop series are: understand the importance of the appropriate TR4 diagnostic for early warning, including field recognition, sample and sampling management, and now the available tools and protocols needed for the TR4 diagnosis. Also, be aware of the significance of carrying out specific surveillance for Fusarium TR4 detection and delimitation, and now the international standards for phytosanitary measures that should be implemented for surveillance purposes. Also, be mindful of the international threat pathways of significant concern for the Fusarium TR4 spread and the commodities that should be subject to inspection. And be better informed of the concept of simulation exercises and understand their usefulness to improve the quarantine pest outbreak response and how to prevent the introduction and spread of quarantine pests such as Fusarium TR4. Next slide, please. The IPPC virtual workshop series on Fusarium TR4 disseminate and communicate essential information that the involved staff in surveillance, diagnosis, and response for Fusarium TR4 should know. The first session on diagnostics charts technical and conceptual information to promote a similar knowledge and understanding, considering the gaps among the countries worldwide. Attendants will receive insights to understand the complexities of the Fusarium TR4 diagnosis, which involves multigenic tests to proceed with a first report. They will be informed on the available and appropriate tools and protocols for TR4 diagnosis, understanding the principles and reasons to select specific methodologies. The virtual sessions represent the more straightforward and efficient way to disseminate this kind of information and knowledge as broadly as possible, and the MPPO responsible staff of the TR4 diagnosis needs to count or develop the required skills to apply the shared knowledge. Next slide, please. For this session, we have the privilege of having global experts, the global expert Claire Quema and Fernando Garcia, who will share relevant and technical information on sampling and diagnosis techniques, followed by a presentation from Mariluz Ayala from the MPPO of Colombia, who will share the Colombian experiences implementing Fusarium TR4 diagnostics. Thank you very much for being here, and I wish you an enjoyable webinar. Please remember that this webinar is being interpreted in French and Spanish, and that's a courtesy from the CELA and Colia CP. Also, please use the question and answers channel to type your questions to the panelists, and the speakers will try to address them in a specific sessions for the Q&A. And we will prepare a document where we will provide the answers to all the questions that are asked, even if we don't have time to answer them today. Now, you will know about the recognition of suspect plants, characterization of Fusarium TR4 symptoms in bananas, and Gerrit Quema delivers this presentation. Gert is a professor of phytopathology and head of the Laboratory of Phytopathology of Wageningen University. He has 40 years of experience in plant pathology, and he is currently involved in several projects and is one of the initiators of the Global Alliance Against TR4 a member of the World Banana Forum, and a member of the IC team on Fusarium TR4. Gerd, the floor is yours. Welcome to this session.
Good morning, good evening, and good afternoon to all of you. Thank you, Camilo, for the introduction, and thanks again also for organizing this virtual seminar. So indeed, my name is Gert Kema. I'm a professor of plant pathology in the University of Wageningen. So my presentation will be mostly on diagnosing and sampling Fusarium wilt in bananas. So let me just share my screen with you and find a presentation. Now it should be full screen. Can you confirm that you see it full screen? Yeah, super.
I can see the full screen. Thank you.
Okay, gracias. All right, so as you can see on this slide, sampling Fusarium in banana, you can see my mouse, I think, as well, right? What do we have here? Let me just choose a laser pointer. You see my laser pointer?
Yes, we can.
Perfect. Okay, so let me just lead you a little bit to Fusarium wilt in bananas with a few introductory slides. So here you see a healthy crop. This is in China, Guangxi. And here on the left, this is a severely affected plant by Fusarium wilt. As you can see, the younger leaves show extensive chlorosis. And of course, that is, once you see this in Cavendish, that is, you should be right away highly alert. But what we have to keep in mind, exactly the same symptoms developed by other race 1 or race 2 strains in other germplasm. And so by just watching the symptoms, we definitely cannot identify whether a strain is TR4, yes or no. And what we have to keep in mind as well is that despite the fact that Cavendish is very susceptible to TR4, there is lots of other banana germplasm that is equally susceptible to TR4. And that's why diagnostics are crucial to timely identify and diagnose whether a particular strain sampled from an affected plant that suffers from from Fusarium wilt is truly tropical race 4, yes or no. But if we focus on Cavendish, and of course Cavendish is about 50% of the global banana production, once you see extensive or starting leaf chlorosis, you should be right away alert and start sampling. Now, you don't have to cut the plant right away, but this plant, once you cut this is the cross-section of the pseudostem, and you see all those affected xylem vessels here. You know, these very dark patches here are heavily colonized, but here in the center, you see a more kind of yellowish discoloration, and that is also a sign for colonization by Fusarium strains. And so, but of course, the initial sample doesn't have to be so drastic, and that's what I will show in the upcoming slides. As I said, banana, very important crop. It's a very important staple crop for millions of people, particularly also in Eastern Africa, but also in Latin America. And of course, it's a very important fruit crop, the top fruit crop of the world. Despite the fact that Cavendish represents about 50% of the global production, there is lots of genetic diversity. But most of that diversity is by, for instance, diploids and fertile diploids. So they're full of seeds, and that's why we don't consume them. And so the triploids are the most important dessert bananas or cooking bananas. Despite this enormous genetic diversity, essentially Cavendish is a global monoculture that really suffers from two very important diseases. Number one is the subject of today, Fusarium wilt. The other one is black sigatoka that requires almost weekly fungicide applications. What's also important to consider is that the far majority of banana growers are smallholders. And of course, there is a big industry, but that represents about, and the export represents about 15% of the global production. But the far majority of growers are smallholders. And to reach out to smallholders, that's essential if we talk about disease management. So this is what I already said. Cavendish represents nowadays more than 50% of global production. And of course, what is important to understand that not all Cavendish is being exported. So only about 15% of the global production is exported. So Cavendish is also very important for domestic markets. Like for instance, in China, they only grow Cavendish. India is a very large Cavendish producer, but hardly export. So that's all for domestic markets. All right, so now and then if you scout from the air, as you can see here in this picture, so this is a piece of land in the Philippines, Mindanao, that is heavily devastated by Fusarium wilt. And so you see those areas within plantations that are, eradicated by the disease. And that's why we really believe that proactive area monitoring is one of the tools that we have available right now to diagnose or to identify suspect areas. Because the fact is, and that's what we have seen throughout the appearance of tropical race 4 from Southeast Asia into the Middle East, the Indian subcontinent, Africa, Latin America, the moment you diagnose it by chlorotic leaves, essentially we are already too late, because it takes really months for the disease to develop, and during that time the disease is already spreading. It's caused by soil-borne fungi, Fusarium species that easily disseminate with contaminated tools, contaminated shoes, boots, water, flooding, storms, of course. And so actually, once you see the chlorotic leaves, we are already too late to really quarantine the pathogen. And so proactive monitoring for suspect areas from the air is key, in my opinion. That's a practice that we have now piloted in Peru with a consortium. And then you can go to those suspect areas, sample plants, and try to be more efficient in monitoring the disease. Now, the history of Fusarium wilt may be good to remind you. So the first Fusarium wilt epidemic was a major epidemic across Central and Latin America, caused by what we now call collectively race 1 strains, and in the then most important variety, Gros Michel, that was really wiped out with a massive effect on, of course, employment for hundreds of thousands of workers, growers. And that was truly a very dramatic event that took place, started about in the 1920s and culminated to this major epidemic in the 50s. During that time, of course, there was a major effort to look for replacements of Gros Michel. And many, many ways to control the disease were implemented. And what you see here on the left picture is inundation. And so inundation, of course, causes an anaerobic condition that suffocates the fungus. Very effective in disease control. The point, however, is that water is also a very good way to disseminate the disease. And in those days, there was no tissue culture. So after replanting these areas, many of those new plants were already infected, and that actually only boomed the epidemic eventually. Now, during that time, they also discovered Cavendish. Cavendish is not the result of a breeding program. Cavendish is a clone that was identified in Indochina, French Indochina most likely, survived barely in botanical gardens, ended up in a botanical garden in the UK, in England, and then survived in Chatsworth House in Derbyshire in the UK. And from there, it was taken to Samoa by Reverend Williams. And there it was found out that actually the Cavendish was resistant to the strains that killed Gros Michel. Now, that was not immediately embraced. That really took, 10, 15 years. But eventually the industry was really pushed towards Cavendish. Many things had to change, in particular the logistic chain. But the key element was that Cavendish was absolutely super resistant to the strains that killed Gros Michel. And it still is. And so that's why we can grow Cavendish on all those soils that are still contaminated with race 1. Once you start planting Gros Michel again there, they will again develop Fusarium wilt within a year time. So the inoculum is still present. Cavendish has shown and is the best possible example of the power of innate resistance and the level of resistance that's required. to really stop the epidemic. So this worked. But then tropical race 4 came up. Tropical race 4 originates from Indonesia, from the center of origin. To make a long story short, it appeared in the 60s in Taiwan, and from there it started to disseminate across Southeast Asia. And then we published in 2014 that it had arrived in Jordan. And since then, the story is in the next slide, and I will summarize that. But this is the effect. This is a Cavendish plantation in Mindanao, 2012. I was there myself, completely eradicated by tropical race 4. So exactly a repetition of history. And this is then the global picture where we are right now. So as I said, initially in the 60s in Taiwan, since then disseminated across Southeast Asia. Then we published it here in 2014 in Jordan, and later on also in other Mediterranean countries. In Pakistan, it was reported in 2018 in India, 2013 in Mozambique. That was the first incursion in Africa. It was even diagnosed in a biome in the UK, and then of course the incursion into Latin America in 2019 in Colombia, and last year in Peru. So we really can talk about a developing pandemic. And despite all the efforts to quarantine the disease, we have to conclude that apparently quarantine didn't work. And so this tropical race 4 strain is now truly disseminated globally. We've also sequenced many of those tropical race 4 isolations from those various countries. And what we can conclude is that tropical race 4 is truly a clonal pathogen. So despite, in contrast to the race 1 strains, which are diverse genetically, tropical race 4 is a single clone. This is the background. So it gives you the full story, more or less, on the origin of the pandemic and where we are at this particular moment of time. Coming back now to affected plants. So here again, you see a plant that is affected by Fusarium wilt. The younger leaves show chlorosis, not as severe as what I showed you previously. And once you observe such a plant, we have to engage in a couple of activities, and that's what I show here. So you go to that infected plant or that suspect plant, take a knife, and you essentially cut out a square from the pseudostem. Make sure you have a clean knife, that you have disinfectants with you, so that you afterwards can clean the knife, use gloves, so take disinfectants along. And once you have cut out this part, that's only, a small part, maybe up to, let's say, 10 by 5 centimeters, and you pull it out, and then you see here some discoloration of the vascular strands, right? And those are the vascular strands that you would like to isolate. That's what I show on the next slides. So this is another example. This was here in Mindanao in the Philippines. Again, a square of about 10 by 5 centimeters. You see here very clearly the discoloration. This is a magnification of this cut here. So you see this is now the material that you would like to collect. So from that part that you have cut out, select those tiny parts of the pseudostem and then dry them. So here you see them. So here we have cut out those discolored vascular strands. You see they are only about, let's say, 10 centimeters, 5 to 10 centimeters long, up to a few millimeters wide. That's really enough because this is heavily colonized by Fusarium strains. Once you have done this, you can take that part that you have cut out, place it back, and just tape the stem. So that is easy to do. And then once you have cut these strands, you fold them in paper. Never do it in plastic, always use paper. Filter paper, newspaper, that really doesn't matter, but put it in paper. And then just let it dry by room temperature. Don't put it in a refrigerator, just by room temperature. Fold the paper, so let it dry for 1 to 2 days. And then it's dry enough, put it in a paper bag, and essentially from there you can just ship this material to the location where it will be further analyzed. What is important in sampling taking is, of course, register all the details of that particular sample. So the variety that it was sampled from, the location, ideally the GPS coordinates, the dates, by whom, and then of course, tabulate the details so that it can be shared with the authorities where you send the samples to. So I just repeat once more, please don't use plastic, just use paper. So as I said, document it carefully. So the cultivar type, the sample type, what part of the leaf of the plant has been sampled, the location, and the date. And that kind of concludes the sampling process. We're only talking about plant samples. We are not talking about soil. The seminar here is focused on taking plant samples. And this is the final slide. And then Fernando, the next speaker, he will go into more detail on how you process then these samples eventually. So once they arrive in the lab, what is then being done? So Camilo, I hope I'm still within the time. This concludes my short seminar on sample taking.
Thank you very much, Gert, for this terrific presentation. I want to invite our attendants to ask your questions into the questions and answers channels. You have got an icon at the bottom part called Q&A. Please use this channel to formulate your questions. Now, as announced by Gert, you will get an overview of the classical and molecular tools for the Fusarium R4T diagnostic in bananas. I have the pleasure to give the floor to Fernando Garcia. Fernando is the head of the banana breeding program at Guijen. He coordinated the reports of the initial incursions of the TR4 in Jordan in 2013, in Pakistan and Lebanon in 2015, in Myanmar, Laos, and Vietnam in 2018, and in Colombia in 2019. Welcome, Fernando. The floor is yours.
Hello, thank you very much. Good morning, good afternoon, probably good night. First of all, thank you for the invitation. I'm very glad to share a little bit of our knowledge with all of you. I'm happy to see many people in the chat from all over the world. That's very exciting. So I will share my screen now. Okay, please let me know if you can see the full screen.
Yes, we can, Fernando.
Okay, perfect. So indeed, we are going to talk about diagnostics today. The title that was assigned to me, quite long title, but in general, we are going to see an overview of the available tools, classical molecular diagnostics, their usefulness, and the minimum tools needed for a first diagnosis on TR4 in bananas. I'm gonna take the laser, yeah. So of course, we cannot talk about diagnostics if we don't know the basics of what are we looking for, right? Like we need to know the symptoms, we need to know how it looks, a little bit about the biology and the genetic of the fungus. Gert gave already a very nice introduction. So some of the slides I'm just going to skip. But essentially, we are going to talk about a little bit of story of the pathogen. Is very important for the taxonomy because recently, as you know, there are suggestions for a new names. So we are going to also see that part. So as Gert mentioned, actually Fusarium Wilt, known also as Panama disease, is one of the most important diseases in the story of agriculture. It was the responsible of the big epidemic on Gros Michel. And it was actually discovered in Australia, but of course, it originated in Southeast Asia. And actually, it was discovered by Dr. Brankov in 1874. So at that time, he didn't know that what was the causal agent of the disease, but he speculated that was a fungus based on his observations in the microscope. So shortly after that time, there were outbreaks in Central America, especially in Costa Rica and Panama, that at that time was actually part of Colombia. So the first reports were same as TR4 in Colombia, and due to the name, to the devastation caused in Panama, it was called as Panama disease. The causative agent was later isolated for the first time by Dr. Erwin Smith, and he got it from samples from Cuba, and that's why he called it Fusarium cubensis. So probably you know, probably you didn't know about why is the name of Fusarium is the TR4 was cubensis for many years. So after a few years, Dr. Brandes was the first person that demonstrated that the Fusarium cubensis at that time was responsible for the disease. And how he proved that, interestingly, he used same techniques that we are using nowadays for the diagnostic. So he inoculated artificially soil, and he reproduced the symptoms in healthy plants. So it's after 100 years, we are using the same concepts. Several decades passed until the German specialist in Fusarium classified as a part of the Fusarium oxysporum complex. Later on, in 1940, thanks to Snyder and Hansen, two scientists who created the Forma specialis in banana, they classified Fusarium oxysporum Forma special cubensis as the responsible for the disease in bananas. Nowadays, we have more studies based on DNA, and that allowed to understand more about the genetics of the fungus, and that also helped with the diagnostics because there are different classifications that we will see later. This was also mentioned. The introduction of Fusarium and the distribution in Latin America was most likely due to the propagation material introduced from other countries, and thanks to the boom of the commercialization of the Gros Michel, rapidly dispersed around the world, and especially in Latin America. So the era Gros Michel was very successful, but it was demised by the presence of Fusarium wilt at that time, race 1. And it was until the shift to Cavendish that the industry of banana was saved. As many of you know, it was not forever. And actually, it's very interesting to know that Cavendish had been showing symptoms of Fusarium wilt for many years. And even from the 20s, some Cavendish bananas show symptoms of Fusarium, especially in the subtropical areas. And that's why we have the race 4, the subtropical race 4. So these Cavendish bananas show symptoms when they are under predisposing conditions, like especially abiotic conditions like winters, inundations. So these are predisposed to be sick for different PCGs associated with the subtropical race 4. However, in the 90s, a little bit earlier, the first big incursions of TR4 were present in plantations in Cavendish, and it was called the tropical race 4 because this time it was present in the tropical areas, and the Cavendish bananas were sick without any predisposing situations. So either in good or bad situations, the Cavendish were always sick. So Professor Randy Ploetz classified that specific strain of Fusarium as a DCG 1213, and later on it was called as the tropical race 4. Fusarium was a local problem in Southeast Asia for many years, and as Gert already mentioned, we've reported for the first time outside of that area in Jordan, and from there, more and more reports exponentially started to appear around the world. And that was actually because of the diagnostics, because we had better tools to make diagnosis faster. Unfortunately, it appeared in Colombia and Mozambique, Islas Mayotte and Peru in the last couple of years. And as I mentioned before, for example, for the case of Jordan, the samples arrived even earlier than 2013, but there were not at that time fast tools to develop a rapid diagnostics. Nowadays, we have better tools to do that. So if we talk a little bit about the pathogen, what you observe now in the screen is an electron microscopy picture of Fusarium. All the green part is the pseudostem of a plant, and you can see that the Fusarium is hyphas are going out of the plant in an advanced state. So Fusarium is a big genera that comprises hundreds of species, especially in the complex of Fusarium oxysporum. It has a lot of different variants like endophytes, saprophytes, and also pathogens. Even Fusarium, there are pathogens that are pathogenic to humans. However, in plants, they are more known because wilting and diseases in roots. Some cases, they also are able to produce mycotoxins, like for example, in cereals, and they are divided in forma specialis. So they are very specific to some crops. For example, as you know, the forma specialis cubense can only infect bananas, but cannot infect peas, for example. And the other way around, the forma specialis species cannot cause disease on bananas, but only on peas. So this is the classification for forma specialis. Additionally to that, we have the physiological races that we already discussed a little bit. In the case of the Fusarium of the banana, is responsible of the Fusarium Wilt or Panama disease. So far, it's been known for many years as Fusarium from forma specialis cubense. However, there is a very interesting study that suggested new classification for different species among this complex of Fusarium that cause disease in banana. So 4 races have been described, and more than 20 compatibility groups have been described. As Hertz mentioned already, contaminates the soils for decades thanks to these specific structures that are called calamidospores that survive under very difficult situations in the soil. There is not a suitable control yet, and it spreads very easily, essentially in 3 ways, by plant material, soil, and water, and everything that you can imagine in between, like soil attached to soils, to tools, to machinery. So those are essentially the 3 ways that how Fusarium moves. Just to summarize here, it's very important for the diagnostics to recognize the races and the VCGs. So in general terms, we can say that the diagnostics for tropical race 4 is kind of easy because the tropical race 4 is comprised by only 1 VCG. So it's a clonal lineage. However, for other races, like for example, subtropical race 4, for race 1, for race 2, there are multiple VCGs associated to that specific race. That makes more complex to develop diagnosis for every individual race, if we talk about the race concept. And there is something more complex, and it's this example that I'm going to give you now. For example, if the Fusarium is isolated from a Cavendish plant, is immediately known as Fusarium subtropical race 4. But as you can see here, if it's isolated from a Gros Michel, the VCG is exactly the same. So these kind of things make this classification very ambiguous and difficult for pathologists. There is another example, the 12-4 is considered in some places subtropical race 4 because can cause disease on Cavendish, but can also be associated with race 1 or even with race 2. So this is a nice, interesting work by Herkema and collaborators in which they describe and confirm the clonality of the Fusarium from tropical race 4. So as you can see, these are isolated TR4 from many different locations and all belong to the same. So they are essentially, it's a clone. It doesn't matter if it comes from Pakistan, from Philippines, from Lebanon, for Jordan, it's just one. That means that it's just one pathogen, one specific strain that is moving in the world. That also points out the responsibility of humans moving the pathogen around. Because as you know, the Fusarium cannot just be distributed by air. So this is the work I mentioned. So there is a new proposal for naming the species that are associated with Fusarium wilt in bananas. But the only thing that I want you to remember from this slide is that recently there are many papers and documents in which you will see this name, Fusarium odoratissimum. Just for you to remember, Fusarium odoratissimum represents what is known so far as Fusarium tropical race 4. That's the only thing that I want you to remember for now. If you want to know more about this work, you can refer to Mariani and read about the new proposal for the naming of different species associated to Panama Disease, to Fusarium wilt. Sorry. The cycle is important also to remember. So everything starts with spores that are present in the soil. They attach to the roots. They can also enter in injures in the roots of the plants and then spread, germinate and colonize and spread to the vessels of the plant. So this is another picture in which you can observe some of the spores of Fusarium. Everything you see on yellow are the micro and microconidias, micro and macroconidias that after just a few minutes are attached to the roots. They germinate and they enter and then colonize and cause what you already saw in Her's presentation, a distribution of hyphas that colonize all the interior. This is a cut of the pseudostem of a plant. As you can see, everything that is like white are the hyphas of Fusarium, colonizing every empty space, also the bundles, the vessels, xylem, blocking the system, the vascular system, causing the wilting, but also breaking into the cells of the plant. And of course, all this colonization of the fungus inside the plant cause the typical yellowing of the plant, the wilting. And in this picture, I'm giving you 3 examples with 3 different cultivars of banana and 3 different races. So you can see Fusarium race 1, race 2, and tropical race 4 in a manzano, an apple banana, plantain, and a Cavendish. As you can see, the symptoms are very similar. And the most important part here, already mentioned by Her, is that in some cases, for example, the TR4 was not detected on time because it was camouflaged into another cultivars. So how do you know if this is not tropical race 4? And that's the importance of the diagnostics, because in this case, we say, okay, it's a plantain showing symptoms. Okay, let's say that it's a race 2, but can be, but it can also not be the race 2, but can be a tropical race 4 that is there, and it will be noticeable and when this jumps into a Cavendish plant. Sometimes you can also see the breaking of the pseudostem, but it's not a common symptom. It's not like 100%, but it happens. And internally, you can observe what Her mentioned before, the discoloration in the vascular bundles, and then these are the samples that Her show you that are collected for diagnostics. Of course, the diagnostics starts in the field and by our eyes, from the air. There are multiple applications that help the human eye to identify, but they are not 100% accurate. So they, like for example, in this case, says that this app says that it's banana corn weevil, but also here Fusarium xanthomonas, the same symptoms. So the symptoms are difficult to identify, so we cannot base our diagnostics in what we observe. Of course, I'm not saying that the molecular techniques are infallible. Actually, they are. You know that there are recent publications that show that there are multiple false positives, and that's why we always recommend a mixture of techniques to be sure. And you will hear me saying that multiple times during this presentation because it's very important. I'm not going to show you how to do the isolation of the fungus, but because it takes a lot of time, but I have some videos on YouTube. If you're interested, you can let me know later on how to do it in detail. Diagnostics are crucial in every step of the management of the strategies for Fusarium. Even before the fungus arrive, and for quarantine and exclusion, it's very important to take samples randomly, to evaluate some plants, asymptomatic plants, symptomatic plants, just to be sure that the Fusarium is not there yet. The same for every step from every strategy of management. Even for I'm now working on breeding, and even for us in breeding, diagnostics are very important because we want to know that the plants that we generate are resistant to tropical race 4 and that they don't have tropical race 4 inside. There is also, when I was working at university back in Her's group, we did a lot of collaboration with multiple groups, and this is just part of the indirect use of the diagnostics by evaluating resistant plants, for example, in scientific works, just to identify if Fusarium is there or not, and also even testing products from companies, if they work or not to kill Fusarium. This is just a little bit of our experience in the diagnostics. This works all together with HERT-KEMA in collaboration with also from ICA in Colombia, in the case of Colombia. So we started in 2000, as I mentioned, 12 and 13, but at that time, it took us a lot of time to diagnose the disease 100%. I'm talking about first reports. So this is more complex. It's not just the first diagnosis, it's really a whole report for first presence of the pathogen in a new country. So as you can see here, there is an evolution of the techniques. In 2000, we started the samples from Jordan in 2013, and only until 2014 we could deliver the result, because we had to do different techniques that were required at that time to have 100% certainty that we have TR4. It was the VCG test, which is laborious and takes a lot of time depending on the number of samples. So later on, we move into the sequencing, and with that, we can do it faster, more or less in 1 month or 1.5 months. Why it takes so long, we will see later. We did, by the way, this for free in collaboration with the University of Wageningen, of course. So the techniques had evolved also a lot. We started from days, even months, to just a few hours and even minutes to give a first diagnosis of the tropical race 4. As I said before, not all the techniques that I'm showing here are 100% sure. So they have some failures, but that's why we recommend to mix techniques to be sure that we have a good diagnostics. So the future probably will be something like this. We are very used to these kind of things nowadays, and probably in the future we will have something like this also for Fusarium. So this is what we did call the first diagnostic or the pre-diagnostic. So techniques that allow you to have a first impression of what you have. If you have tropical race 4, you have to start immediately with the, like Herb said, an alert, especially if it's in Cavendish. So I've been working with many types of samples, so plant material, fungus itself, soil samples, and with that, together with Herb, we evaluated many samples around the world as positive. And of course, this gave me a lot of experience, and thanks to that, I had the opportunity to travel, to teach, to train people in Latin America. This is a picture of our last workshop in Latin America, which involved people from Bolivia, Peru, Ecuador, and Colombia. And thanks to the Comunidad Andina who sponsored this workshop. So with that, we wrote a nice guide. Unfortunately, it's only in Spanish, but we are looking for collaboration to translate it into other languages. So this is a complete guide that includes from sampling to first report steps, talking about soil samples, also everything related with the LAMP technology, which gives the results very fast, and how the work in Colombia was done to identify for the first time Fusarium tropical race 4 there, including the system. So in the diagnostics, we always work with the Koch postulates. So even though these postulates were developed for human diseases, many plant pathologists adopted this because it gives a complete round of diagnostics that will guarantee that the report you deliver is 100% sure. So when we talk about pre-diagnostics, we are talking about all these techniques that allow you to have a first impression if you have a positive yes or not. But until that point, you can have false positives. So when you have, especially for first reports, where countries free of Fusarium that show plants with Fusarium, then you have to go to the second stage, which is the pathogenicity tests and sequencing or VCGs. So the pathogenicity tests simply make reference to isolate the fungus that you isolate from your suspicious sample. You grow that fungus, you infect healthy plants, and then you isolate again the pathogen from those artificially inoculated plants, and then you have to confirm again that is the same one that you used from the beginning. So you start with this, go to the pathogenicity test, and return to this just to confirm once more that it's the same. So Herb already showed you the steps of the sampling. These are examples from sampling in Colombia, from plant, but also from soil in a work in collaboration with ICA a few years ago. And here I'm going to show you the steps for the isolation. So we get the samples, and then these are These are going to vary a lot because depending on where you do the diagnostics and in which country, in which lab. So when you get the samples and depending on which kind of sample you have to process those samples, you can do it manually, as in many countries. In other universities or institutions, you probably have more sophisticated tools. The same goes for the DNA isolation. You can do it with the kits, or you can do it with reagents prepared at home. Or also in some places we have robots that do it for us. Quantification the same. There are multiple options for this kind of evaluations and different techniques. So in this case, you can choose. There is not big difference regarding which kind of tool you're going to use to process the sample, because I know I'm from Colombia and I know what are the difference between the Netherlands and Colombia, for example, limitation in infrastructure, in machinery, etc. But what you cannot play too much around is with selecting which kind of primers are you going to use. So far we have three options. We have traditional PCR in real time, and the LAMP. So as mentioned before, I recommend always to mix at least two of these samples to give a diagnosis. So when we use the first one, the TR4 by DITA 2010, you can do it directly into the plant and also in isolated fungus, DNA from pure cultures. We have the primers developed by Carvajal based on the 6 genes, which they have a pipeline to describe the disease, and it has an extra step of restriction, but it works well. And we have another primers from China, which work also well. But we recommend to mix, because sometimes some of these amplify for not only tropical race 4, but also for subtropical race 4. And that's very important, because just imagine if you give a report saying that it's tropical race 4, but resulted to be subtropical race 4, because remember that the subtropical race 4 can also affect Cavendish bananas in certain circumstances. So it's very important to use different techniques. Very important, use special controls for your DNA. So we have several in the guide. We recommend one for Fusarium oxysporum, for Fusarium cubense, for fungal in general, and also for DNA for banana. So we also have different techniques with PCR, with real time, and the LAMP, which is very good and works very fast. So the previous one is for a first report, but when you have a first incursion, so let's say your country is free of TR4 and you have suspicious plants, then you have to do the complete round of diagnostic, including what I just mentioned, one of the techniques of PCR, qPCR LAMP, or also additionally, you have to use a VCG test or the genome sequencing, and not sequence of the fragment, because if you sequence the fragment, it's essentially the same as doing a normal PCR. Then the pathogenicity test to prove that the fungus that you prove as positive will produce the same symptoms as they observed at the initial point. And then you have to go to another PCR round just to confirm that the fungus you have is the same as the one that you isolate at the beginning. This is very important. The proper use of these methodologies depends on the skill of accuracy of the individuals making the diagnosis, which is critical to prevent false positives, or what is even worse, the false negative. So this is the diagnosis I use, but of course I recommend it, but there are multiple options. So we receive the samples, we take let's say a plant tissue, then we extract the DNA, and at the same time, we also isolate the fungus from that plant tissue. We extract the DNA, and then we perform one round of PCR, qPCR, or LAMP using at least 2 different techniques. Then here we will have a positive, and that will be the first alert. So with this, we inform immediately the owner of the sample that we might have Fusarium. However, if it's negative, that still has the possibility to be a positive, because when you extract DNA from the plant, most of the DNA is from the plant and not from the pathogen. So it has to be very sensitive technique, like for example, LAMP, to catch a little bit of DNA of the fungus into the plant tissue sample. But if you go to the re-isolation of the fungus, then you extract the DNA exclusively of the fungus, then it's easier for all of these techniques to detect a positive. Then if we have a second positive here, it's important to tell that this tier 4 diagnostic scheme is for samples that come from Cavendish, because then you already know that there is something special there. Then when you have the second positive here, you go to the pathogenicity test and either sequencing or VCG test, and then you have a third positive sign. And this is what we call a complete diagnostics for a first report. So it has to have all these 3 checkpoints. If it's negative here, remember that the plant's from a Cavendish. So we have to pay special attention to these samples. If it's negative after this, still very interesting. So we have to repeat it because we need to be sure that that sample that comes from that Cavendish is not a false positive, false negative. So we need to go here. And then if we have a positive, we go to sequencing and pathogenicity test. We also get samples from soil. So we start here, re-isolation, and we also get fungus sometimes directly. So in paper filter, so we go directly here. So that's that was my presentation. Just some final remarks. So the current pandemic is caused by a single clone of Fusarium known as Fusarium tropical race 4, represented by the VCG 12, 13, 16. So diagnostics are crucial at every stage of disease management. So it's not just to define if there is a new incursion, but it has to be involved in every step of the management of even the prevention. The effectiveness of control measures is still very weak, even though all the things that are being done, Fusarium, we know by story that Fusarium will continue to spread. We recommend a combination of oligos and techniques to give accurate diagnostics and prevent the false positives and false negatives. Everyone with proper equipment and training can do diagnostics. And something very important also to point out, the topic that Gert mentioned is that once you see Cavendish infected in the field, it's too late. So even we can prove that this tropical race 4, like for example, when we identified in Colombia in July, more or less, that doesn't mean that the Fusarium arrived in July. That means that Fusarium was there at least. Being optimistic, it was there at least for 1 year before it was, because you know, it takes some time for Fusarium to express the symptoms in the plant. So the moment we see it in the field, it's too late. It's been there already for at least 1 year, and saying it optimistically, at least 1 year. So with that, I would like to thank everyone, especially the organizers, Gert from Wageningen University, and Mariluz from ICA, because we have had a lot of collaborations in the past few years. Thank you very much.
Thanks, Fernando, for this complete and helpful information on the available tools on Fusarium tier for diagnostics and on the proper techniques for this purpose. You delivered significant insights to manage a field report of Fusarium tier 4, and I hope this information be useful for all our attendances. We already have several questions in the questions and answers channel, but I wish you to animate you to provide more questions. So please do not hesitate to formulate the questions in the Q&A button and note in the chat on the right. Now we will have a 10-minute break, and we will launch through the chat an evaluation survey of this webinar. It will take you less than one minute to complete it, and you will help us to improve our future events. Please fill this questionnaire, and we will share the link through the chat. So please follow the link, and please, once you follow the link, select the language of your choice to select English, French, or Spanish. It's just less than 1 minute to provide us your your feedback. So let's have a short stop, and then we will continue with the presentation of Mary Luz Ayala from the National Plant Protection Organization of Colombia.
Pour moi, c'est ce qui est en train de se passer. Et ce que je vois, c'est qu'il y a toute une série de pays, quand même, que vous preniez le Moyen-Orient, que vous preniez l'Inde, que vous preniez la Chine, et qui semblent être en train de dire, écoutez, la civilisation que l'Occident nous propose, on n'en veut pas. On n'en veut pas, toute cette histoire. Donc, il y a une espèce, c'est peut-être un truc civilisationnel qui est en train de se passer.
Thank you to those people who already answered the survey. Until now, we have 53 responses, 64 responses. So we would like to increase this number and encourage all of the attendances to respond this webinar evaluation survey. We have reached 102 answers. Thank you very much for all your support answering this webinar evaluation survey. We will have a couple of minutes more to wait for your answers, and then we will start again with the next presentation. Some people is informing through the chat that the link is failing, so we will send it again to try to facilitate you access the link. If you had any difficulties accessing the webinar survey evaluation, please try again through the last link available in the chat. Thank you very much for all your support. Let's continue with this webinar session. Thank you again for filling out the webinar survey. In the meantime, keep in mind that we will be answering as many questions as possible, either in written form or orally during this webinar. But after this webinar, we will also share a document with all the answers on our International Phytosanitary portal on the webinar website. Okay, thanks again to our friends who answered the evaluation survey. We reached almost 153 answers, and thank you, it's appreciated your support. Now, let's continue with this webinar session. We will stop the break. I hope all of you are ready to start this webinar session. Our next speaker is Mariluz Ayala. The National Plant Protection Organization of Colombia will share with us their experiences in the implementation of Fusarium R4T diagnostic in Colombia. The presentation, as I mentioned, will be delivered by Mariluz Ayala. Mariluz is the technical lead of the Fusarium R4 diagnostic at the MPPO of Colombia, that is the Instituto Colombiano Agropecuario. She has extensive experience in plant pathogen research and diagnosing diseases of agricultural importance. And just to acknowledge that it is almost 6 a.m. In the morning for Mariluz, where she is located. So thank you, Mariluz, for joining this workshop session. And the floor is yours to start as your preference.
Buenos días para todos. El agradecimiento es para ustedes. La invitación a compartir en este espacio las experiencias que desde el ICA hemos tenido es muy importante, es muy importante para el país. Aspectos positivos, aciertos o errores que digamos, ustedes puedan reconocer en nuestra experiencia, igual resultan beneficiosos para que los puedan incorporar en procesos similares que tengan en sus países. Entonces, como digo, más que nada agradecer pues, a la PAO por la invitación. Les voy a compartir mi pantalla. Me confirman, por favor, si la están viendo.
Yes, we can.
Perdón. Como les decía, la idea entonces es compartir la experiencia y los desafíos que hemos tenido en Colombia tras el surgimiento de FOC R4T en el país para, digamos, facilitar, digamos, el recuento de la historia, aunque es una historia reciente y corta, pues hay que digamos, de diferenciar como 3 momentos en este proceso. Tuvimos inicialmente en el 2016 y 2017 un proceso de estandarización del protocolo de detección para FOC R4T. Como decía Fernando, lo mencionaba ahora, pues digamos que todo el desarrollo de metodologías de detección permitieron que en organizaciones de protección fitosanitaria como nosotros ya pudieran pensar y estructurar un protocolo de detección. Nosotros trabajamos, como les digo, en el 2016 y 17 en este proceso. Ya en el 2019, entonces, tenemos esta introducción o esta identificación del patógeno en el país. Se genera el primer reporte para Colombia y pues para el continente. Realmente, aunque digamos que la dispersión de los patógenos y en el caso de POC ya era algo inminente, creo que ningún país, por lo menos de Sudamérica, esperaba ser el primero. Y fuimos nosotros, y luego a partir de esto, obviamente, se halla todo el desarrollo de una serie de actividades para mejorar los procesos de diagnóstico. Obviamente que nosotros teníamos que comenzar a pensar en atender las demandas crecientes del diagnóstico que se iban a generar dentro del país por la intensificación de la vigilancia que un primer reporte generaba. Entonces, básicamente en ese proceso de estandarización, que es algo, digamos, que debemos destacar, en el país, digamos que se trabajó a nivel interinstitucional, participaron diferentes sectores. Obviamente que el renglón del banano es de gran interés nacional, tiene una importancia, pues, digamos, económica y también de seguridad alimentaria. Entonces, con estos intereses, entonces se logró hacer un trabajo interinstitucional. Participaron, digamos, el sector de investigación, también gremios y, por supuesto, todo esto liderado por el Instituto Colombiano Agropecuario. Muy importante, y esto aprovecho la oportunidad para agradecer en todo este proceso, desde esa estandarización que se hizo desde el 2016, tuvimos el acompañamiento del Banana Research Group, liderado por el doctor Quema y en cabeza de Fernando, quien nos acompañó en un proceso de entrenamiento. Y bueno, el acompañamiento realmente ha sido continuo. Entonces valga esta oportunidad para agradecerles a ellos dos que nos han llevado de la mano, y es muy importante de verdad que la academia y que la investigación haga, digamos, este acompañamiento a las organizaciones de protección fitosanitaria, que muchas veces, digamos, no tienen esa misión de investigación. Ya en el 2016 y 2017, entonces nosotros iniciamos todos los procesos de estandarización de los métodos en diferentes laboratorios del ICA. En ese sentido, pues nosotros en ese momento logramos diseñar una metodología para la que se estableció un abordaje de acuerdo a los materiales de banano. Es decir, pues digamos que la manifestación de síntomas en estos diferentes materiales, entonces obviamente que generaban diferentes alertas, y en ese sentido, por ejemplo, para los materiales de tipo Cavendish que se pudieran presentar con sintomatologías, entonces teníamos como una estructura de diagnóstico un poco más robusta. Como mencionaba Fernando, lo voy a nombrar mucho acá porque él ya hizo pues toda esa introducción en la parte de diagnóstica, y como les digo, también nos ha acompañado mucho. Entonces, en esa línea del diseño del diagnóstico, entonces se consideraba la parte de análisis a partir de tejido vegetal, que lo que buscaba era, si se llegaba a este tipo de muestras a los laboratorios, poder generar una alerta temprana, pero necesariamente se tenía que dar una confirmación del diagnóstico a través de los aislamientos que surgieran de esas muestras. En este punto, nosotros en nuestro protocolo, este protocolo se basaba en un único marcador molecular, el IgS, región intergénica, pero digamos que por lo menos se abordaba, digamos, como les digo, desde estos dos tipos de matrices, desde tejido vegetal y micelio. Tenía varias fases, las diferentes fases, obviamente que había una forma de aseguramiento de la calidad que también es muy importante para las ONPF, y con esto digamos que nosotros cubríamos la rutina de análisis de muestras. En el 2019, entonces, digamos, llegan a los laboratorios muestras de Cavendish con síntomas muy sospechosos y son procedentes de la región norte del país, de La Guajira específicamente. Con esto los laboratorios pues ya, digamos, con una sospecha cierta porque hay un material Cavendish con unos síntomas característicos, se realizan pues los procedimientos que nosotros teníamos definidos para ese momento, esos procedimientos de diagnóstico, y con eso digamos que obtenemos unos resultados positivos a partir de tejido, y obviamente que se obtienen, digamos, los aislamientos a los que se les hacen los análisis correspondientes. Con esto generamos ya una alerta y un resultado sospechoso. ¿Por qué no confirmarlo, digamos, con estos resultados del laboratorio? Pues porque se trataba precisamente de un primer reporte para el país y para el continente. Entonces era necesario tomar, digamos, tener todas las garantías. Una cosa importante es que el diagnóstico desde las organizaciones de protección fitosanitaria va más allá de las técnicas. Obviamente, eso lo vamos a mencionar más adelante, hay unas implicaciones, digamos, legales, sancionatorias. Entonces, obviamente que hay que tomarse, hay que tener y ofrecer todas las garantías en estos procesos analíticos, y en este sentido, digamos que lo que se decidió desde la institución, igual, digamos, es algo que se aplica en todos los países que hacen nuevos reportes de FOC R4T, obviamente que tratan de hacer un diagnóstico, un proceso de diagnóstico, digamos, más complejo, completo, como lo explicó también Fernando anteriormente. Y entonces vuelve la Universidad de Wageningen a través del doctor Fernando García apoyarnos en este proceso de confirmación. Entonces, las muestras son enviadas a sus laboratorios, donde se realizan unos procesos adicionales con los que ya pues podemos confirmar y tener ese primer reporte oficial para el país. Básicamente, digamos que las estrategias diagnósticas y las técnicas que se utilizaron para esa confirmación fue un proceso de secuenciación masiva, test de patogenicidad, obviamente, y técnicas adicionales como LAMP. La idea era, pues, obviamente tener, digamos, marcadores moleculares adicionales que pudieran, como les digo anteriormente, garantizar la confiabilidad, pues, de esos resultados, sobre todo para el alcance que tenía ese primer reporte. En este sentido, vale la pena mencionar técnicas como BCGs, esas evaluaciones no fueron utilizadas. Estos procesos demandan mucho tiempo, pero además que los otros procedimientos empleados, digamos que complementaban muy bien todo este proceso de diagnóstico, entonces en este caso no fue utilizado. Bajo estos resultados, entonces, ya se produce ese primer reporte oficial en Colombia en el 2019. Lamentablemente, pues ya como también mencionaban, en el año pasado, otros focos fueron identificados en Perú. Esto pues es, digamos que, lamentable para la región, pero lo que simplemente genera es, digamos, una necesidad de trabajar, de insistir y trabajar mucho más en esto del manejo y control de POC R4T. En ese mismo sentido, y entonces con el problema en el país, entonces desde el área de diagnóstico, nosotros tenemos que pensar en nuestros aspectos, como les decía, el diagnóstico en las organizaciones de protección fitosanitaria va más allá de las técnicas, y el ICA como tal, digamos que es la organización que tiene que, digamos, garantizar la fitosanidad en el país. Obviamente elabora unos planes de manejo y control de la enfermedad. En el caso de POC R4T, esas estrategias de manejo se basan, digamos, de forma general en toda una serie de medidas de bioseguridad que tienen que estar acompañadas pues como con esta parte del diagnóstico. Entonces, esa bioseguridad que están implementando en campo, nosotros en los laboratorios tenemos que tratar pues como también de implementarla, ¿cierto? No puede ser que los procesos de diagnóstico, de análisis en el laboratorio vayan en otra vía. Obviamente que las actividades en el laboratorio, que muchas veces están relacionadas con el aumento de los inóculos, toda esta parte de movilización de muestras hacia los laboratorios genera unos riesgos importantes para la dispersión del hongo. Entonces, obviamente que en el laboratorio, desde el laboratorio también nos teníamos que ocupar de esa parte importante, y ir en esa misma vía de todos los planes de manejo y control de la enfermedad. Entonces, en ese sentido, se plantearon como 3 líneas de trabajo en el laboratorio. Inicialmente, en el proceso de diagnóstico, sabíamos que teníamos que fortalecer nuestra metodología, nuestro método analítico, porque en ese mismo momento en que se da el primer reporte en el 2019, también sale una investigación donde se reportan problemas de falsos positivos y negativos con el marcador precisamente que nosotros estábamos utilizando. Entonces, lo que decidimos es reforzar, digamos, en el método analítico, incorporar nuevos marcadores, ¿cierto? Que le aportaran una mayor confiabilidad a nuestros resultados. También tendríamos que trabajar como en los tiempos de respuesta de esos procesos de análisis, y en el mismo sentido teníamos que pensar siempre que esos procesos de diagnóstico, de detección, tenían que tener todas las garantías de calidad, y en ese sentido, en esa misma línea, pues nosotros comenzamos a fortalecer ese sistema de calidad que tenemos en los laboratorios. Obviamente que el ICA siempre, y los laboratorios siempre han trabajado con este sistema de calidad. Viene pues ya un trabajo muy anterior al reporte de la enfermedad, pero ya digamos que bajo esta situación sabíamos que teníamos que poner mucho más la lupa ahí, trabajar mucho más. Otra línea, entonces, en la que teníamos revisar era sobre el incremento de la capacidad analítica. Nosotros, para el momento en que se registró ese primer foco, en que tuvimos ese primer reporte, teníamos 2 laboratorios habilitados para el análisis de FOC R4T, lo que se hacía insuficiente porque se sobrevenía un incremento en las muestras que iban a llegar al laboratorio, porque se intensificaban pues las labores de vigilancia. Entonces, en eso también teníamos que pensar. Y por otro lado, también en la bioseguridad que íbamos a implementar para nuestros procesos de detección. Como les digo, las actividades del laboratorio no puedan ir en contravía de los planes de manejo en campo. Entonces, la idea con esto de la línea de bioseguridad era tratar al máximo de minimizar los riesgos de dispersión asociados a la movilización de las muestras. Y en ese mismo sentido, en el laboratorio, digamos, asegurar la contención del patógeno que potencialmente se iba a manipular. Como mencioné anteriormente, el control oficial va más allá de los protocolos de detección, entonces nosotros tenemos que cuidar otros aspectos adicionales, los resultados que nosotros entregamos a los usuarios, que en este momento son básicamente internos, o sea, son de los proyectos de las dependencias de la institución que realizan la vigilancia. Esos resultados pues son utilizados para tomar decisiones relacionadas con el manejo, por supuesto, pero muchas veces también acciones legales y procesos sancionatorios, que finalmente todo esto redunda en la protección fitosanitaria pues como del país. Entonces, para esto, como les decía, el sistema de calidad se cuida mucho más en el proceso específico del análisis de FOC R4T, y nosotros obviamente que ahora estamos trabajando en reforzar toda la parte pues como de metrología asociada al proceso, el equipamiento, digamos que la formación del personal dentro de los laboratorios, los sistemas de aseguramiento de la calidad, los materiales de referencia. Digamos que en eso insistimos bastante. Por otro lado, sobre el proceso de detección que les hablaba, pues que una de las cosas era que teníamos que hacer algunos cambios para robustecer esa metodología a la luz pues como de pronto de reportes de falsos positivos sobre los marcadores que veníamos utilizando. La parte inicial, que es muy general, esa digamos que permaneció igual con el proceso de diagnóstico que veníamos trabajando. Digamos que el doctor Kema y Fernando ya mencionaron, digamos que algunos detalles sobre ese proceso que son generales de las técnicas de análisis en el laboratorio, la importancia de tener una buena muestra. Los resultados analíticos nunca van a ser mejores que la muestra que nos llevan, entonces cuidar esas características de la muestra dentro del laboratorio son súper importantes. En ese mismo sentido, pues el proceso de aislamiento y los de extracción, valga mencionar acá que nosotros para las extracciones de ADN utilizamos un protocolo paso a paso, no utilizamos metodologías con kits comerciales. Este protocolo que implementamos nosotros nos ofrece bastantes resultados, además digamos que por temas, digamos, presupuestales también ofrece importantes ventajas. En el esquema general, entonces, que establecimos ya para el análisis de las muestras, ya pues digamos que con el patógeno presente, ya teníamos como un esquema general para abordar todo el tipo de muestras independientemente de los materiales de musáceas que nos pudieran llegar. Entonces simplemente definimos que para muestras asintomáticas íbamos a utilizar de pronto la técnica más sensible disponible, que hasta ese momento era una QPCR de un kit comercial de la empresa Clear Detections, que también se basa en la región IGS. Y por otro lado, para las muestras sintomáticas, entonces teníamos todas las dos líneas de análisis a partir del tejido y obviamente en el caso de obtener, digamos, aislamientos fúngicos, obviamente estos aislamientos fúngicos se procesaban y para el análisis de los aislamientos fúngicos, entonces incorporamos un nuevo marcador, que es básicamente lo que les decía. En este caso decidimos incorporar los genes 6, el análisis de genes 6, que se basa básicamente en una técnica de punto final. El doctor Fernando pues había proyectado ya este esquema diseñado por Carvallis en el 2019, digamos es un esquema bastante robusto porque a través del diseño que se propone se pueden identificar varias razas, en el caso de nosotros pues de interés. Es digamos la confirmación de la identificación de raza 4 que tiene implícito un proceso de restricción enzimática, ¿cierto? Para la confirmación de raza 4 tropical. Entonces, en este momento nosotros tenemos esto en nuestro procedimiento. Acá un poco relacionamos, digamos, todos los primers, todos los marcadores moleculares que utilizamos dentro de nuestro proceso analítico y recalco acá que pues la idea es que en cada fase del proceso de detección se incluya, digamos, un aseguramiento de la calidad. Lo que les digo es que es muy importante en todo esto del diagnóstico oficial ofrecer sobre el resultado que se emite al usuario la mayor confiabilidad, todas las garantías en este proceso. Entonces, es muy importante que cada fase, en la medida de las posibilidades, tenga incorporado un sistema de aseguramiento de la calidad. Entonces, para el análisis de tejido, obviamente que se analiza, que tenemos un control interno para ese ADN de planta y en el mismo sentido, para el proceso de análisis de micelio, nosotros de forma general realizamos una amplificación de genes y TS para asegurar, digamos, que la calidad de esas extracciones. Acá hay un esquema, perdón, más gráfico de lo que hacemos en el laboratorio en nuestro proceso de detección a partir de tejido. Entonces, realizamos un análisis de IGS, ya sea por la metodología duplex de DITA o bien con el kit de Clear Detections. De forma general utilizamos el kit de Clear Detections, ¿cierto? Que es, digamos, mucho más sensible por utilizar pues como la técnica de QPCR. Y en la otra línea está entonces el análisis para en micelio, los aislamientos fúngicos obtenidos. Hay una, digamos, etapa dentro de este proceso de análisis de aislamientos fúngicos, que es la identificación de la forma especial. Nosotros, digamos que esta parte la decidimos incorporar en el proceso, digamos que tiene varias utilidades. Puede funcionar indirectamente como un control de calidad sobre el micelio. Cuando, digamos, el hongo efectivamente corresponde a la forma especial cubense, pero si no, también digamos que para fines prácticos esto de alguna forma aporta información para, digamos, las dependencias del ICA que hacen la vigilancia de, digamos que de otras razas que estén presentes en el territorio. Entonces, digamos que por eso se decide, digamos, dejar dentro del proceso del análisis al nivel de la forma especial. Y acá, obviamente, que también se hace el análisis del hongo con IGS y, pues, digamos que de forma complementaria, el análisis con los genes 6 que les mencionaba, que tiene un proceso posterior de restricción enzimática para confirmar ya pues como la identidad del patógeno. En este camino, nosotros hemos reconocido como la importancia de tener siempre en el laboratorio disponibles como técnicas en el caso en el que se sucedan de pronto ambigüedades sobre los resultados. Es muy importante que el laboratorio con sus herramientas, digamos, pueda garantizar, digamos, un resultado concluyente sobre el proceso de detección. Entonces, en ese mismo sentido, nosotros también tenemos unas técnicas adicionales que siguen pues en estudio. Estos procesos de detección dentro del laboratorio pueden ser muy dinámicos. Obviamente, como mencionaba también el doctor Fernando, pues no hay técnicas infalibles y pues la biología también es dinámica. Entonces, un marcador que hoy puede ser, digamos, muy eficiente, de pronto en un tiempo no sea así. Entonces, sí es importante tener, digamos, diferentes herramientas para, como les digo, garantizar esos resultados concluyentes. Estamos entonces, a veces alternativamente utilizamos una QPCR diseñada por Aguayo para la identificación también pues como de FOC R4T. Este ensayo, precisamente este marcador, funciona óptimamente sobre ADN obtenido a partir de tejido vegetal. Otras actividades adicionales, digamos que en las que ha participado el ICA, que ha realizado pensando en toda esta importancia pues como el diagnóstico para el manejo de la enfermedad, ha sido por ejemplo la participación en un proyecto regional de la comunidad andina para la estandarización del diagnóstico de FOC R4T. Entonces, en este proyecto se plantearon varias fases, algunas de ellas ya se han ejecutado. Se realizó un taller también regional que lideró el doctor Fernando García. Muy positivo este tipo de actividades que sirven para hacer esos intercambios de experiencia y pues y precisamente cumplir con el objetivo del proyecto, que es nivelar esas capacidades. También dentro del proyecto se concibió la estandarización de protocolos para los países miembros, pues como de la comunidad, y la comunidad realizó un aporte. Los países decidían, pues digamos que sobre qué técnica o sobre qué protocolo tenían que trabajar. En el caso específico del ICA, nosotros abordamos la técnica del AMP. Como le digo, siempre estamos trabajando en tener estrategias adicionales que nos permitan, digamos, mejorar los procesos de detección. Y finalmente también dentro del proyecto está considerado, en este momento está en desarrollo, un ensayo de aptitud que nos permita también verificar esa competencia técnica de los laboratorios de la región. Obviamente que este esfuerzo que se hace para, digamos, el control, el manejo de la enfermedad, ese esfuerzo se se multiplica cuando se hace, digamos, de forma interinstitucional, cuando todos los países, digamos, que trabajan en el mismo sentido. Entonces, esto, digamos, es bastante positivo. De este proyecto hasta ahora tuvimos un producto que también lo mencionó Fernando, y es esta guía. Me parece a mí que este tipo, digamos, que de iniciativas y documentos para los fines de los laboratorios son bastante aportantes. Es para un analista, pues, digamos, de cualquier laboratorio, resulta muy ventajoso tener consolidado en un documento toda la información de algún patógeno de interés. Entonces, digamos que eso fue bastante importante. En la parte de la AMP, digamos que con esto del proyecto y que el ICA escogió para continuar también este proceso de estandarización, entonces estamos adelantando estas actividades, reconociendo en la técnica un potencial muy grande para el diagnóstico in situ, que también de cierta forma nos permitiría también ampliar esas capacidades de análisis dentro de la institución. Trabajos adicionales con otros sectores, con otras instituciones también se han venido realizando. Con Agrosavia, digamos que es la institución en el país dedicada a la investigación del sector agropecuario. Se han desarrollado varios proyectos, se trabajan en varias líneas. Obviamente que una de las líneas que se cubren ahí es que se incluyen ahí es la línea de diagnóstico, y Agrosavia ha hecho un trabajo en el último tiempo tratando de desarrollar nuevos marcadores para el análisis de FOC R4T. Nosotros en este momento, entonces, también estamos en un proceso de evaluar la eficiencia de esos marcadores. La idea es tratar, pues, en estos procesos de detección, la idea es llegar en algún momento a un método depurado que pueda, pues, ofrecer obviamente unas garantías importantes en la confiabilidad. Entonces, hacia allá vamos. Revisar siempre, ese es un poco el trabajo desde el área de diagnóstico de las ONPF, revisar qué hay disponible dentro de la investigación, dentro de la academia, que nosotros podamos adaptar y que nos pueda servir, que pueda aportar de la mejor forma a los procesos que se realizan desde la ONPF relacionados con la inspección, vigilancia y control de enfermedades. También se está trabajando en el desarrollo de métodos de detección en suelo. Digamos, esa es una solicitud importante que sea identificado a nivel de gremios. Esa matriz, específicamente la de suelo, es una matriz bastante compleja. Se está revisando a ver cuál sería el alcance de un proceso de detección de esos, porque, pues, como les he mencionado ya bastantes veces, lo que se debe asegurar desde los laboratorios de una organización de protección fitosanitaria es ofrecer al usuario la mayor confiabilidad y garantías sobre los resultados. Por otro lado, en este trabajo que se ha hecho, que se ha generado después de ese primer reporte, entonces, en lo referente a la ampliación de la capacidad analítica de la institución, entonces nosotros pasamos de tener en el 2017 2 laboratorios autorizados que prestaban el servicio para la detección de FOC R4T. Pasamos de 2 laboratorios a tener en el 2021 ya 6 laboratorios que están autorizados para prestar este servicio. Algo importante, no fue concebido, digamos, directamente para la vigilancia de foco R4T, pero fue un proceso que se vino desarrollando paralelamente. Fue, digamos, la adecuación de un laboratorio móvil de diagnóstico agropecuario para el ICA. Entonces, en este sentido, nosotros vemos acá un potencial también para ampliar las capacidades institucionales. Si bien acá hay una cosa para tener muy presente y es todo lo relacionado con bioseguridad, pues están pensando en algunas estrategias para que este laboratorio de forma biosegura pueda también apoyar los procesos de análisis de este patógeno. Es un laboratorio que tiene pues como toda la dotación en equipos, pues, se pueden implementar dentro de técnicas básicas de microbiología, pero también moleculares. Entonces, nosotros esperamos que con esta nueva adquisición, pues digamos que la capacidad del ICA mejore. Ahora, por el otro lado de la bioseguridad, entonces, para fortalecer esa parte, como les decía, nosotros desde los laboratorios no nos podemos permitir ir en la otra línea o afectar todas esas estrategias de control que se hacen en la enfermedad. Entonces, definimos que era muy importante establecer protocolos para la toma, el empaque y el envío de muestras a los laboratorios. La movilización de las muestras aumenta esos riesgos de dispersión. Entonces, para eso se definieron protocolos. También para el envío de las muestras, para el redireccionamiento de las muestras a los laboratorios de acuerdo al riesgo de las mismas y a las áreas muestradas. Les voy a explicar ahorita como más gráficamente ese punto. Lo otro era definir en los laboratorios áreas específicas para la manipulación del patógeno. Nosotros las hemos denominado pues como áreas de contención que permitieran o que permitan el manejo más bioseguro del patógeno dentro de los laboratorios. Y lo otro finalmente era diseñar protocolos específicos relacionados con la manipulación de las muestras en el laboratorio y con toda la parte del proceso analítico de FOC R4T y por supuesto con la disposición final de los residuos que digamos pueden representar un foco importante de dispersión del patógeno si no se manejan adecuadamente. Entonces, de forma más gráfica, tenemos como 3 niveles de contención que siempre tenemos muy presentes para, digamos, realizar cualquier actividad en el laboratorio. La fase inicial, que es la relacionada con el movimiento de muestras, y es esa delimitación o el direccionamiento de las muestras según los laboratorios, con lo que les digo, con los protocolos de envío de muestras. La segunda fase sería la contención del patógeno del laboratorio. Si bien en este momento todos los laboratorios no tienen áreas construidas específicamente, sí en los laboratorios que ahorita realizan los análisis hay una área exclusiva para la manipulación del patógeno, y un proyecto que estamos desarrollando este año es finalizar esas adecuaciones. Y finalmente la parte de los protocolos de bioseguridad para la manipulación de las muestras y los desechos, que considera, por ejemplo, una implementación adicional y diferencial de, por ejemplo, indumentaria dentro del laboratorio. Se refuerzan ese tipo de prácticas en los laboratorios de diagnóstico fitosanitario de forma general. Digamos, no somos tan rigurosos con ese tipo de protocolos, pero para el patógeno, para este patógeno precisamente, entonces se decidió que esas prácticas deberían ser más rigurosas. El uso, por ejemplo, de elementos desechables también para la manipulación de la muestra, que digamos disminuye los riesgos de pronto en el lavado de materiales donde pueden quedar impregnados con propágulos del patógeno. Entonces también decidimos incorporar ese tipo de elementos a los procesos y lo que les decía a disposición final. Entonces, en el sentido del direccionamiento de muestras, lo que les decía, tenemos nosotros 6 laboratorios habilitados, 2 en la zona norte del país y otros 4 ya pues como en el interior del país. Las muestras procedentes de la zona norte, que son las potencialmente positivas en este momento, entonces son redireccionadas hacia el centro del país, donde tenemos unos laboratorios que tienen unas condiciones muy específicas, pues como de aislamiento. Uno es el laboratorio, por ejemplo, de cuarentena vegetal, que es un laboratorio que está un poco más aislado, no está dentro de zonas productoras o zonas productoras importantes por lo menos. Y los otros ya se distribuyen porque valga aclarar que la vigilancia, digamos, se han intensificado de tal manera que se realiza monitoreo a lo largo y ancho de todo el país. Inclusive, por ejemplo, en el archipiélago de San Andrés se toman muestras para ejercer esa vigilancia. Y en campo, obviamente, como les digo, entonces están todas las medidas de bioseguridad para la toma de muestras y envío a los laboratorios. Una cosa importante es que para ese envío de las muestras al laboratorio se tuvieron que reforzar algunos aspectos, pues porque obviamente hay que garantizar que esa custodia sea segura y responsable, disminuir riesgos, por ejemplo, de pérdida de muestras. En Colombia un poco la geografía es difícil, hay zonas de difícil acceso, entonces es importante garantizar que en ese proceso de movilización de las muestras no vamos a tener como riesgos muy altos de pronto de la dispersión del patógeno por alguna pérdida o que una muestra quede donde no debería. Por otro lado, lo que les decía relacionado con los laboratorios, entonces ya hay laboratorios, digamos que se concibieron específicamente para ese manejo de patógenos que requieren un alto nivel de bioseguridad. Entonces, en uno de nuestros laboratorios, digamos, se pudieron construir sistemas de pass-through para ingresar las muestras al interior de los laboratorios, reduciendo pues como riesgos ahí en la manipulación. También tienen un acceso restringido, que es muy importante, restringir el acceso a esas áreas. Lo que les decía, hay todo un sistema pues como de protocolos de bioseguridad con el uso de la indumentaria. Utilizamos una indumentaria completa para la manipulación de estas muestras. Las muestras solo se manipulan dentro de cabinas de bioseguridad y dentro de estas áreas hay unas medidas adicionales para eliminar o no ingresar pues como ciertos elementos. Lo del uso de materiales desechables, que es súper importante. Y finalmente, ya estoy acá por terminar, les quería también mostrar que como un desarrollo paralelo, nosotros en los laboratorios, con todo esto de FOC R4T en el laboratorio se tienen proyectos para la evaluación de resistencia de materiales. Entonces, para el ingreso de estos materiales, nosotros también hemos trabajado en el diseño de una plataforma diagnóstica en la que se evalúan patógenos cuarentenarios para el país. Entonces, digamos que esto también dentro del proceso es, digamos, algo muy importante y un logro en la línea del diagnóstico. Esto es lo que tenía para compartirles hoy y bueno, agradezco, como les decía inicialmente, el espacio.
Marilu, thank you very much for your excellent and complete presentation. It was our last presentation, and now we can move to a question and answer session. Almost of the questions have been answered through the question and answer channel, so it let us the space to give the floor to a couple of participants that are raising the hand. So we will proceed to tell the participant that we are giving the floor. We will start with Rosanna Abdullah. Could please open your mic, Rosanna, and provide your question. Only one question is admitted for participant. Please provide your question, Rosanna. Can you hear us, Rosanna? Okay, it seems that it's not available, so let's move to Joanna Fonseca, who have a question. So open your mic, Rosanna. Giovanna, sorry. It seems that they are not ready to formulate the question, so let's move to Angelo Nunez. Please open your mic to formulate your question. It seems that it's not ready to formulate the question. Let's move to Dimberu Shumi.
Okay. It seems that something is happening with the people that is asking the floor. Yeah, Eva, can you hear me?
Yes, Camilo, I can hear you perfectly.
Oh. The headset is disconnected, sorry. So it seems that they are not ready to formulate the questions. So
to discuss just a couple of questions that were provided through the question and answer channel. The first question would be from Maha Alusabi, that is asking if there's any way to differentiate from the infection by Fusarium type based on the symptoms. I would like to invite Gert Kemat to answer this question.
Sorry. Yeah, sure. Yeah, I answered the question, I think, already in the box. So the question was, can you repeat the question just real quick?
Is there any way to differentiate from the infection by Fusarium type based on the symptoms?
No, impossible. And so, as I said, once you see Fusarium Wilt symptoms in Cavendish, you should be super alert, because that's most likely caused by TR4, although we have subtropical race 4. But the frequency of TR4 infections in Cavendish is simply very higher. So that should be really lead to an alert situation. But TR4 can also cause Fusarium Wilt in many other banana accessions. Race 1 can also affect other banana accessions, and by the symptoms you cannot differentiate race 1, race 2, or TR4 infections.
Thank you, Gert. We are going to recap on some questions from the attendants in the Q&A channel, just to provide more clarity or to extend information for all the participants. The next question is from Kappa Alu, who is asking that apart from drying vascular tissues or sample on tissue or paper at room temperature prior to sending off to the lab diagnosis, can we desiccate the tissues in calcium chloride or silica gel?
Yeah, no problem.
Thank you, Gert. The next question is for Fernando. It's a question regarding the availability of kits for diagnosis. Jamarin Elubarin is asking if there is available a rapid test to detect Fusarium TR4?
Okay, thank you for the question. As I think this is more a question for Gert. Of course, for traditional conventional PCR, the primers are publicly available, so it's a matter of order them. The kit is also available. There is one kit, Clear Detections, which works very well. And for the lamp, I think that's a question for Gert, because it's mostly developed at the University of Wageningen. I'm not completely sure if it's already available for everyone, but I think if I'm correct, contacting Gert, he can provide the suppliers and give access to that.
Would you like to add something about the lamp developed by Wageningen University, Gert?
Yes, I can do that. Sorry, you can hear me? Yeah, you hear me, yeah?
Yeah.
Yeah, sure. So the paper was published. I have to check the details. So you can check the details from that paper. If you would like to use LAMP, just contact me at my email address, because we have a group inside Wageningen University that is enabling access to the equipment and the primers necessary for LAMP. Fernando mentioned, of course, the Clear Detections kit, right? So that's the commercially available kit. Maybe what I would like to underscore, and maybe opinions differ here, but what I like in a commercial kit is really the advantage that everybody is using the same protocol. And that makes the comparison of data really possible. Whereas differently published primers, our experience is that in many cases protocols are being changed, and that really reduces the reliability. And that's of course an advantage of a commercial kit, which is, standard kit used can be used by anybody. But Fernando also mentioned, I think, and correct me if I'm wrong, Fernando, but it's always good to have a dual test. And I think that's the advantage also of the LAMP kit, because the LAMP kit is also based on another genomic region in the Fusarium genome. So if one kit is positive, the LAMP kit should be positive as well.
Exactly. And also, if I may add something, during the training that we provide last year, we compile all these available techniques and we test them, because there is a publication recently that claims the false positives, which we confirm actually in the lab that is true. That some of the primers give false positives with some endophytes, but so far the LAMP worked well and it didn't give any false positives. So it confirmed that it's a reliable technique so far.
Thank you, Gert and Fernando. The next question is addressed to Mariluz Ayala. Adelaide Fernandez is asking, what culture media do you use to the fungus isolation? Mariluz.
Sí, me escuchan, qué pena. No, el medio realmente es un medio general, un medio de PDA con cloranfenicol, con un antibiótico. Realmente no hay, digamos que, mayor complejidad en ese proceso.
Okay, Mariluz, thank you for your answer. It seems that we've reached the end of today's session. Almost all the questions have been answered to the Q&A channel. So thank you for following the presentation, which will be posted on the web page of the webinar, together with the recording of today's session. We reached almost 60 answers, with almost 160 answers of the evaluation questionnaire. Please fill it out before leaving this session. The link is available in the chat section, and we will appreciate if you could dedicate a minute of your time to respond to the survey before. I want to thank all our panelists, Gerd, Fernando, Mariluz, for all your kind support to develop this first session, to deliver this first session. Your presentations were excellent and were pretty useful. Also, thanks to the COMESA Secretariat for the support in the execution of the COMESA project. And I want also to acknowledge the support from the COLEA-CP, which provided the French interpretation, and acknowledge the Secretariat of the Simultaneous of the SELA, the Economic System of Latin America, which provided a Spanish interpretation during this session. So once again, thanks for having attended this webinar session. Thanks again for our panelists. Please register for the 2 upcoming IPPC webinar sessions on Fusarium Tier 4, which will complement this webinar. The second session of this series will be on 19 April and will cover the surveillance issues. Also to note that the IPPC Secretariat will deliver certificates to the attendants that complete all the 3 sessions and approve a simple questionnaire. And after that, we will provide you with that certificate. Thank you very much again, and have a nice rest of the day, night, or afternoon. Thank you very much.
Thank you.
Yeah, thanks and bye-bye.
Muchas gracias y hasta la próxima a todos.